I am Dr. T. Virdagiri, Professor and Head of Department of Chemical Engineering, Annamala University. First of all, I would like to welcome all of you for this lecture on fermentation technology. Fermentation is a very interesting word, means the liquid boiling. Why the fermentation processes all are exothermic processes, which simultaneous evaluation of carbon dioxide takes place. That's why the fermentation has been coined. It is a biological process involving microorganisms or enzymes as a catalyst. In fermentations, we have many types of fermentation processes. Aerobic fermentation processes, anaerobic fermentation processes, and facultative fermentation processes. Aerobic fermentation processes, in which the microorganisms are biocatalysts, which requires air or oxygen for its growth and also for its metabolism. Anaerobic fermentation processes, in which the microbes are anaerobic in nature, that means it does not require air or oxygen for its growth or for its metabolism. And facultative fermentations is a combination of maybe aerobic or maybe anaerobic type. The organisms may grow in presence or in absence of air or oxygen. Sometimes the first phase we may have to carry out in presence of air, and the second phase, product formation phase, essentially we have to carry out in absence of oxygen. So a combination is desired in some of the cases. So the present world is biotechnology. Bharat tomorrow is biotechnology. So many of the products are aimed to produce through biotechnology. Because of its simplicity, because of its ease of operation, because of its operating lower temperature, pressure, and other operating conditions are normal room temperature and atmospheric pressure, and the equipments used are very minimal, and that's why the cost is very minimum. And biotechnology process offers very huge advantages over chemical processes. The cost of catalyst involved is very less. The cost of chemical catalyst is very enormous and cannot be compared, and only we have the contamination problem in biological process. That's why in pharmaceutical industries, antibiotics production, and food processing industries, many biochemical industries all go for fermentation products, and a lot of profit is made out of these processes. And insulin, interferon, which are mainly required for diseases like diabetes and cancer, all those things can be, these products can be obtained by fermentation processes economically. That's why this fermentation technology, of course, is a new program, but one can learn from the products made in the home itself. The products made in the home, namely the curd, the idli, and other products, if one can observe thoroughly, they will be able to understand what is fermentation. The fermentation, during the fermentation processes, the biochemicals, various biochemicals are added, the medium is prepared, the medium is sterilized, and after sterilization of the medium, the fermentation is carried out in presence of microorganisms. So naturally it is an exothermic process with the evolution of carbon dioxide. It depends on important factors like temperature, pH, and then aeration, agitation, and all those factors govern for the economic efficient fermentation. In a normal house, you might have observed the curd, we don't get curd during the winter season because of the cold climate and low temperature process. At the same time, at the hot summer seasons, we get curd very easily, and very fast fermentation takes place. Likewise, every factor can be analyzed, and fermentation should be designed appropriately. Fermentations can be classified into microbial fermentation and enzyme fermentation. The microbial fermentations employs various types of microorganisms: bacteria, blue-green algae, fungi, protozoa, and other organisms. Whereas enzyme fermentation employs many enzymes, protease, lipase, glucoamylase, many enzymes are involved in enzymatic fermentations. Enzymatic fermentations are costlier because of the cost involved in the enzymes, whereas microbial fermentations are cheaper because of the cost of the microorganisms which acts as catalysts are much cheaper and can be produced in the industry itself. Only problem with the microbial fermentation is the contamination problem. Most of the fermentation products are produced in batch wise because of the larger residence time required. The enzyme fermentations, the quality of the product is better. Further, the fermentation processes can be classified into simple fermentations, simultaneous fermentations, and then stage-wise fermentations. In simple fermentations, there is a direct relationship exists between the rate of product formation and the rate of carbohydrate utilization. And the simultaneous product formation, more than one reaction takes place simultaneously with different stoichiometric ratios on conversion of raw materials into products. There is no direct relationship between the carbohydrate utilization and the product formation. And the third category is the stage-wise production. There is no direct relationship between the rate of product formation and the rate of carbohydrate utilization. So similarly, the classification of fermentations will give a better knowledge in understanding the fermentation process. So in fermentations, first of all, one has to select a suitable organisms which can produce the required fermentation product. So the fermentation, the organism chosen should give maximum product yield, and required medium which is economical for commercial production should be formed. The medium essentially consists of carbon source, nitrogen source, energy source, the mineral source, and vitamins. The carbohydrate source essentially consists of starch and then molasses, cellulosic materials, and all types of carbohydrates, which is essential, and all the process calculations are based on the carbohydrate conversion. And the nitrogen source includes various nitrogen sources like urea, ammonium sulfate, ammonium nitrate, sodium nitrate, and various other nitrogen containing compounds, and biological compounds like soya bean seeds, lentils, and this beef extract, yeast extract. All those things are the good nitrogen sources. So a medium essentially should contain a carbohydrate source, a nitrogen source, an energy source. In most of the cases, the carbohydrate source and the energy source are same. Sometimes we have to add some extra substance as energy source, otherwise there is no need for it. And minerals, all types of minerals like calcium, magnesium, potassium, strontium, cobalt, nickel, all those essential elements should be added in trace quantities. The microorganisms are very sensitive. Even if one element is absent, even though it is required in micrograms, it will not grow. So for example, the Lactobacillus leishmaniae, a culture used to analyze the vitamin B12 in microbial assay, doesn't grow in absence of vitamin B12. So very small quantity of vitamin B12 is must in addition to all other ingredients for its growth. So similarly, cobalt and other elements also is very much essential for its growth, but is required in very less quantity. And the other factors affecting the fermentation, as I already told, are temperature, pH, aeration, agitation. The temperature, effect of temperature on the rate of fermentation, I would like to say, when you increase the temperature, the rate of fermentation processes increases very fast, and we get a maximum yield at a particular temperature, and then afterwards the yield decreases with the increase in temperature. That is due to the death of the cells. The cells cannot withstand at high temperatures above 40 degrees Celsius. Most of the organisms will survive between 30 and 40 degrees Celsius. Above 40 degrees Celsius, the culture growth rate gets affected and cells starts dying, and hence the rate of production gets reduced. And then temperature is further increased, the yield becomes less and less. When you operate at a very low temperature, the yield is very less, and we have to adopt an optimum temperature for maximum product yield, whether it is microorganism or enzymes. Enzymes, in case of enzymes, enzyme denaturation takes place at high temperatures, and that's why the product yield becomes less and quality becomes less. When you use pH, similarly, we can have an optimum pH. Most of the microorganisms prefer to grow at an optimum pH of 7. At low pH, very less microorganisms prefer to grow, and some microorganisms prefer to grow at very high pH ranges. Most of the cultures grow at pH 7. We have advantages if our culture is in the range of acidic or in the range of alkaline pH. If it is in the range of acidic range or alkaline range, the contamination problem is naturally overcome, which is normally expected in all fermentations. To some extent, we can avoid the contamination problem by choosing the cultures which can grow in acidic pH or in alkaline pH. In acidic pH, most of the cultures prefer to grow at the pH 7. So I already mentioned about temperature. Temperature, you can effect of temperature. I would like to mention the organisms can be classified into psychrophilic, mesophilic, and thermophilic organisms. The psychrophilic means low temperature the organisms grow. Mesophilic means the temperature is between 20 and 40 degrees Celsius. Then thermophilic means above 45 degrees Celsius. You can call the organisms as thermophilic organisms. The energy source for most of the microorganisms in fermentations is same as that of the carbon source, any carbohydrate materials. Only very few cases we add extra chemical substances as energy source, and in very few cases the energy source is an important factor for most of the fermentations. The energy source is in most of the fermentations is same as that of the carbon source. In some cases we add extra chemicals as energy source for fermentations. In some cases the carbon dioxide serves as an energy source for algae and other photosynthetic organisms. Photosynthetic organisms play a vital role in effluent treatment also. And then mineral substances are an important factor in all fermentations. Mineral substances, calcium, potassium, all those things has to be added in very trace quantities, and fermentations can further be classified into growth associated fermentation and non-growth associated fermentation. For proper design of fermentations, one should know the type of fermentation, growth associated fermentation or non-growth associated fermentation. Growth associated fermentation, the product is formed along with the biomass. Both biomass, which is also considered to be one of the bioproducts, biomass and the products are formed simultaneously with consumption of the substrate, namely carbohydrates. In non-growth associated product, first the growth takes place. The product formation takes place. In growth phase, the microorganisms takes all the substrate and nutrients and then grows, and then it attains the maximum growth, and later on the product formation takes place. When you consider the growth cycle of microorganisms, there are 4 important phases. One is the lag phase, the other one is the log phase, and stationary phase and declining phase. These are the 4 phases of growth cycle of microorganism. The knowledge about growth cycle is important for all types of fermentation, because when we have to harvest the microorganism, when we have to inoculate the culture into the fermentation is more important. So the lag phase is defined when a culture is inoculated into a new medium. The culture takes some time to adjust with the new environment, the temperature, the pH, aeration, agitation, and the composition of the medium. It has to adjust, then only the growth takes place. The minimal time period required for is preferred. The culture which requires minimum period, minimum lag period is preferred for most of the fermentations. And the next period is lag period. Immediately after the lag period, the log period starts, or the exponential period starts. The exponential period, where the maximum growth occurs, the growth rate is much greater than the death rate. Where the substrate is being consumed, the product, namely the biomass formation takes place. The yield of biomass increases as time of fermentation increases. Then it reaches a maximum biomass concentration. At the same time, the substrate concentration falls to a very minimum, beyond which the substrate cannot be consumed. And then the maximum, then after reaching the maximum biomass, the concentration remains same. That phase is called stationary phase. Then the stationary phase, the growth rate as well as the product formation rate are equal. The growth rate and the death rate are equal. And then after some time, there is no energy source available in the medium, and hence the declining phase is inevitable and is caused due to the much faster death rate than the growth rate. So for fermentations, we need organisms belonging to exponential phase. The exponential phase culture is very active cultures. This has to be added to the fermentations. And if a culture is harvested in a stationary phase or declining phase, we won't be having a very active fermentations. So the cultures harvested during the exponential phase are preferred, and we should first of all study the growth cycle of each and every culture. We should try to develop new cultures which can produce maximum amount of products. So the by mutation technique, one has to develop newer cultures. For example, in olden days, vitamin B12 was cost lakhs of rupees per kg, but now it is available in a minimum cost. Similarly, insulin, interferon, all chemicals are available at low cost. And so the proper technology is important. Fermentation technology, proper operating conditions, proper mutants, high yield mutants should be developed. So the now our researchers have ventured into bacteria which can desulfurize, the oil very efficiently. In our laboratory, carried out research and they have been awarded national award by AICTE director for best MTech thesis. So this lot of researchers on ethanol production, on production of various biochemicals, citric acid, oxalic acid, and many other fermentation products are being carried out in our laboratory. This fermentation technology can also be applied to wastewater treatment also. In wastewater treatment, particularly for anaerobic digestion. Anaerobic digestion is one of the important process in which a huge quantity of energy is derived. Pharmaceutical industries, many biochemical industries which leave lot of solid residues, liquid effluents, can go for this anaerobic digestion process, wherein all the residues, all the waste materials are dumped into a huge digester, particularly distilleries and then pharmaceuticals. They dump all the waste into a huge anaerobic digester, digest them. Only the disadvantage is the slow process, where we can get huge quantity of biogas, which is nothing but 55 to 65% methane and rest carbon dioxide, which is used as an energy fuel, energy and particularly as a fuel, and for lighting purpose also it is used. There is a huge biogas plant available in Punjab, which caters to the need of the entire village. The electricity, the fuel for each and every family is supplied from a huge biogas plant. And we have also a huge biogas plant in our agriculture faculty. And anaerobic digestion offers more advantages in producing their energy needs in industries, also in production of steam and power also. It has been tried for the various cogeneration systems also have been tried. Among the other important factors which affect fermentation are aeration, agitation, and dissolved oxygen concentration, etc. This aeration, when I say aeration depends on the type of organisms used. The entire fermentation may be aerobic. You have to supply air continuously, and then if it is anaerobic, we should not provide air. Complete anaerobic condition must be provided. For aerobic condition also, the optimum quantity of air flow rate must be designed and provided for proper growth of microorganism, for proper product formation. So if sometimes the fermentation may be a combination of aerobic and anaerobic fermentations. In those cases, the first phase is aerobic, means the vitamin B12 fermentation consists of 7 days. In the first 4 days, maybe aerobic growth maybe is preferred, and this next 3 days is anaerobic condition may favor the product formation. So we have to thoroughly study the fermentation and provide suitably the aerobic or anaerobic conditions to get maximum product yield. Agitation also plays an important role. Agitation, if we go for a very high RPM agitation, this may disrupt the cells. The cell walls may be disrupted, growth rate gets affected, the product yield becomes less and less. And then the RPM is very less, means that the agitation becomes less. The mass transfer aspects are very poor. Mass transfer and heat transfer aspects are very poor. So the transfer of oxygen in most of the fermentations from bulk gas, that is air, to the medium becomes difficult, because if it is a fungal fermentation like used in penicillin fermentations, this transfer of oxygen from the gas to the liquid medium becomes difficult. So we should use proper horsepower of the motor, proper impeller design, proper agitator design is important, and we have to supply sufficient power for mixing. Mixing is essential for growth as well as for better mass transfer of oxygen, to provide uniform concentration throughout the reactor. And then aeration, agitation, and then dissolved oxygen concentration. Dissolved oxygen concentration is another important parameter which we have to provide sufficient dissolved oxygen concentration. That is by supplying air only, again, by providing air in sufficient quantity. When in all fermentations, the type of reactors play an important role. Most of the fermentations we carry out in batch reactors because of the larger residence time. The culture growth period is a very larger, because it requires larger residence time. The batch reactors are preferred over continuous reactors. Most of the fermentation products are produced by batch reactors. In continuous reactors, very few products are produced industrially. Continuous reactors are aimed to increase or to have a very high yield of products, to increase the high yield of products, and to make the process completely automation. In continuous processes, the contamination problem, possible mutations are disadvantages. Mutation chances are more in continuous processes, and providing larger residence time is difficult in continuous fermentation tanks. In a batch fermenter, the simple fermenter which include the stirring facility, facility for measurement of temperature, facility for measurement of pH, aeration, agitation, and dissolved oxygen, redox potential. Baffles are provided in the fermenter. Simultaneous sterilization facility is provided in the fermenters. Fermenters, we can study lot of things in the production of biochemicals. In controlled atmosphere, we can produce enzymes. we can produce biochemicals, we can produce antibiotics, we can produce a number of fine chemicals. So this batch fermenter, we charge all the feed together into the fermenter, we sterilize them after adjusting the pH, and then we inoculate with the required culture. Inoculation is important factor in all fermentations. Inoculum with the required size, and required size is important. Inoculum required size and age is important. The age refers to the culture at which it has been harvested. So the culture age and size plays an important role in successful fermentations. So the fermentation medium with all essential components has to be inoculated at a proper stage, and we have to get it fermented for the required period of time, and then once the products are formed, the product separation becomes difficult. So the product separation, we call it as downstream processing. The downstream processing only is the area which consumes a lot of cost in fermentation technology. The downstream processing includes the absorption, adsorption, liquid-liquid extraction, all membrane extraction processes, reverse osmosis, electrodialysis, all membrane separation processes. So most of the cost in fermentation processes goes for these steps, namely separation of the products from the fermented broth, not on the production stage. The production, up to production stage, the cost involved is very minimal. So this downstream processing has also to be well planned and well designed. Actually, the design of reactor, proper design of reactor is an important factor. This, as I already told, in batch reactor we charge the reactants, we get them fermented, and then they separate the products. In continuous bioreactors, the feed is continuously passed through the biocatalyst. The biocatalyst may be an immobilized bed containing the microorganisms fixed onto a inert carrier, or the enzymes fixed to an inert carrier. These things are used in the continuous packed bed reactor, or fluidized bed reactor, or bubble column reactors, or airlift fermenters to get the substrate converted into product. Now the continuous fermentations are all in trial stages. Only very few continuous fermentations are successful. Now most of the scientists go for fed batch fermentations because they wanted to provide an uniform substrate concentration throughout the fermentation period, because the microorganisms, which are biocatalysts, attains the stationary phase because of the non-availability of the substrate, and then the growth stops thereby, and then the product formation also gets reduced. So to increase the growth rate continuously, the substrate or the other nutrients also fed to the bioreactor continuously at a particular rate to maintain uniform concentration. Thereby the growth rate as well as the product formation rate are enhanced, and the product yield is increased. So the proper design of reactors, either fed batch or continuous reactors, are an important factor in design of all fermentation. In brief, fermentation technology is going to replace almost all chemical processes in successful production of commercial products, maybe ethanol, methanol, or propanol, or vitamins, or antibiotics, or any products. Because of the cheaper cost involved in it, because of the cheaper equipment cost, the total profit available is more. So this fermentation technology is a very simple technology which involved the very less cost chemicals. The operating conditions are quite normal, and then everything is very favorable conditions when compared to chemical processes. When compared to chemical processes, the catalyst cost is less. The fermentation period is more, and the yield is maximum. The catalyst poisoning is not there, and the fermentation plays a very significant role in production of many pharmaceutical products. In brief, the fermentation technology is going to replace most of the chemical processes. Because of the lower catalyst cost and the cost of chemicals and other maintenance cost, operating cost are less in case of fermentation processes. In chemical process, the catalyst cost is more, catalyst poisoning is there. And then for successful fermentations, we have to design the fermentation medium, optimum components, their compositions, their sterilization methods, and then inoculation procedures, inoculation time, the time of fermentation, everything should be properly designed. We have to adopt a suitable reactor for maximum product yield, either batch reactor or continuous reactor or fed batch reactor or bubble column.